Hello, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today for what is my favorite episode of the year. No offense to any of the other amazing guests that we have, but we're back for a second time with Dr. Veera Rajagopal, who's a scientist at Regeneron Pharmaceuticals and is famous for his tweet threads. He's at Dr. Veera on Twitter, and he also has a Substack with longer form stories called GWAS Stories, and I really recommend you follow him on Twitter and subscribe if you're interested in this kind of thing. But he has an incredible knowledge of all the latest and greatest in genetics and drug discovery. And if you remember our episode from last year, we covered some of the biggest stories of 2022, and now we're back again in 2023, and it has been a really interesting and amazing year. So Veera, first of all, thanks for agreeing to come back, and I'm really looking forward to the discussion.
Thank you so much, Patrick. So I think I should start by saying that I'm a big fan of genetics podcast. I've been listening to most of it. It's incredibly informative. So it's an honor to be here again, and thank you for the invite. Looking forward to our discussion of 2023 exciting Human Genetics Roundup.
Yeah, it really is my pleasure. I think one of the great things about having this podcast, I get to talk to people who are have just incredible knowledge of the field, and I get to learn by osmosis. And for you, this has been a big year. You published quite a significant paper that I suspect you probably worked on for a number of years, looking at a rare variant involved in smoking or lack thereof. Maybe you can talk a little bit about that. And you designed a cover page, which has a beautiful illustration of DNA helix turning into a puff of smoke. Did you use the AI Midjourney or Dolly or something to design it, or do you have artistic talents as well?
Yeah, thank you. Thank you for mentioning that. Yeah, so the cover page, yeah, I designed it myself. So there's no AI assistance. So I used to do work on Illustrator and Photoshop during my med school times, and as well one of the go-to person to design brochures, college invitations. So it's kind of a hobby. It's been long time since I used it, and we had this idea, why don't we suggest a cover for Nature Genetics? And then I was trying some commercial illustrators, but time was very short, and then just a weekend I thought maybe I should give it a shot, and it just worked out. So I'm very happy. I'm kind of more excited about the cover page than the actual paper getting published.
I thought the paper was very interesting too. Maybe you can talk a little bit about it, because it's related to a lot of what we're going to discuss today, which is how both rare and common genetic variants can really provide a window into biology and drug target discovery. So maybe you can talk a little bit about that.
Yeah, absolutely. So, I guess we had this. I introduced myself even during the last podcast, what we do, what the kind of work we do at Regeneron. So we are mainly in the drug target discovery area, so where we use human genetics to identify drug targets. And one of the very specific types of associations that we are interested in is protective associations. And so I'm working in the neurology, psychiatry, addiction fields. And this is one of the first project that I started. I joined Regeneron in middle of 2021. And yeah, so it was very excited to find this. It's not a very novel association, but it's the first time we are getting a human genetics validation of the target. It's actually a subunit of nicotine acetylcholine receptor. CHRNB2 is the name of the gene. And this encodes a subunit of the most predominant receptor, nicotine receptor in the brain. And this is a known target of Chantix, varenicline, that's been first line drug used in the smoking cessation. And so we found the human genetic evidence, and it was very exciting because the Chantix was designed based on another chemical molecule called cytosine, which goes really long back, even before we know about all these nicotine receptors. So I think it's like a serendipitous flavor of drugs where you identify these molecules even before knowing how the mechanisms act. And retrospectively, we kind of have this human genetic validation, which also very well aligned with the mice genetics. It's one of the landmark paper from Marina Picciotto, and they are the pioneers in this nicotine receptor biology. So it all worked well. So everything aligned perfectly. And this is kind of the main highlight is this is the first behavioral, like genetics drug targets that has been identified. So, like always there's criticism about doing genetics for behavioral phenotypes. And, it was like, we was very excited to show this even for, behavioral phenotypes where it is really difficult to do genetic studies. We can identify biologically meaningful targets like this.
And how does this, for those who haven't read the paper yet, how does the mechanism actually work here? So I think carrying the variant was associated with about a twofold decrease or 35% decrease in likelihood of smoking heavily. What is it that's going on in the brain of people who carry this rare knockout or rare loss of function of this particular gene?
Yeah, so it's a good question. So basically the main finding here is we find that individuals who carry rare deleterious mutations in CHRNB2 are protected from smoking heavily. And also we see this protection at multiple, like disease indications that are downstream of smoking. And so this kind of validates the mechanism of varenicline, which actually is a partial agonist antagonist of this nicotine alpha 4 beta 2 receptor. So this is the main switch, if you can, you can say, for nicotine to act on the brain. And having a deleterious mutation kind of switches off this, and so nicotine cannot act. So it's mainly cutting the rewarding effect. So we believe that people who have mutations smoke, they won't feel as much rewarding effects as in non-carrier. So this kind of protects them from becoming addicted to smoking.
Interesting. How much is known about the drivers behind other kinds of addictions, whether it's alcohol or gambling? Do we have because this is a direct kind of chemical link to the nicotine, and I'm wondering whether there are master regulators of addictive behavior and those sorts of things, or whether they tend to be on a compound by compound level, if that makes sense.
Yeah, that's also a great question. So there is at some level, there is some kind of common, like there is convergence in the molecular mechanism. So the midbrain region actually have this reward neural circuits, right? So, and this neural circuit is like kind of believed to play a role in different types of addiction. Like it can be cannabis or smoking, or even can be like, like internet, or, like gambling addiction, and even some related to, like obesity. So this reward pathways are mainly to be blamed, but the genes and, like there are probably like sub-biological pathways that are kind of have more specificity towards each indication. So smoking, nicotine receptors, but we don't know, like we really just scratching the surface of what nicotine receptors, like involved in the brain. There is like a huge amount of literature. And then for alcohol, we have, like opioid systems, but also we have the, alcohol enzyme in the liver. That's one of the mechanism of disulfiram, which inhibits the alcohol processing enzyme. So yeah, there are like compound specific stories, but also in a general, there is like some kind of common biology behind multiple addiction. And I think like we are still learning. We are in a very early stage of understanding them.
Yeah, and the part of the reason I was wondering was there are anecdotal reports, and I think probably trials either ongoing or coming, looking at some of the GLP-1 agonist, Ozempic, generic name I think is semaglutide, but many people are saying they use this to lose weight, but actually see impacts on other kinds of addictions, or I should say addictions, not that the losing weight is or having to lose weight in the first place is the result of an addiction. But yeah, I was curious there where there's clearly a mechanism there that needs to be investigated a little further on this one.
Yeah, that is something I'm also like very keenly, watching. So there's like multiple when the Ozempic thing was like, was like everyone talking about it a few months ago. One of the things that was really stood out for me is that like people, like serendipitously reporting also some kind of protection against smoking and alcoholism. And this really took up. So I think like Paul Kenny, a very big shot in the field of addiction biology from Mount Sinai. he even made an announcement in TV about the clinical trials they're starting this
Right.
year or next year for like this GLP-1 receptor agonist for addiction indications. And yeah, it's really, really great. And hopefully, there might be some genetics also will emerge. We'll see. So it's something to watch for. Yeah, it's very
Yeah.
exciting. And if it turns out, then I think already it's a big thing. So it's going to be a huge thing.
Yes, absolutely. And maybe that'll be one of our 2024 stories if somebody cracks that case. But one of the big, very exciting stories that is actually only maybe a week or two old 3 weeks old by the time we release this episode, but approval of Casgevy, which is, I think, the first successful translation from an initial discovery from a genome-wide association study rather than a rare variant or founder population type of study all the way through to a therapy, and it's an approved therapy for sickle cell disease. Now, I wonder if you could talk a little bit about that. You've got it as your most exciting story of the year. I think I'd probably have to agree with you, but maybe you can talk a little bit more about what's going on there.
Yeah, definitely. So for me, this is without any doubt, the most exciting story of this year, like because, I'm more interested in the GWAS literature and stuff. And this is one of the very early discoveries made in 2007. And this is the first successful translation of a GWAS finding into a drug target. So that's like makes this 2023 landmark in the GWAS history. And also, like, I think it's important that we also need to highlight the genetic aspect of it, right? So, I mean, this approval marks the success of two technologies. One is CRISPR technology. That's what most of the people are talking about. And, like some of us genetists is also like try to shout out about hey, this is also like, this BCL11A was first discovered through GWAS studies. So I was like very into this sickle cell disease and this literature. I've been digging into this. I've been also talking to some of the people involved in this, like Vijay Shankaran from Harvard. He is one of the main person involved in, decoding all this hemoglobin, fetal hemoglobin switching and everything. There's so much, but maybe I can just, like highlight just a few things about it. So first is, I think, how this drug acts, right? So,
Yes.
so basically they use a CRISPR system to make a double stranded nick at a region, which is basically the enhancer region for BCL11A. And this results in reduced expression of BCL11A, which is the main transcription factor responsible for fetal hemoglobin to adult hemoglobin switching, which happens, after birth. And so this results in increased fetal hemoglobin production, and this helps both sickle cell disease patients as well as beta thalassemia patients. It's more striking in sickle cell patients where it reduces the vaso-occlusive episodes and also pain. Pain is the worst thing clinical future. Really, the pain they suffer is really like cannot imagine. So I think that's where it works really great. And so that's how the drug works. So the rationale for HbF induction to treat sickle cell disease. So sickle cell disease is caused by a point mutation in the beta globin gene. So one of the first molecular disease that's been like identified. And this results in, uh, less, like reduces the solubility of the hemoglobin, and it just gives this specific, the polymerizes the hemoglobin molecules, resulting in specific sickle cell shape, right? So one of the earliest finding that was like, observed in the natural history study and also, from the clinical observations is that patients with sickle cell mutations, they do not have, like symptoms, newborn do not have symptoms in the first few months after birth. And it was hypothesized that this could be because the hemoglobin switching is still happening. So there's like high level of fetal hemoglobin. And that stimulated natural history study. Then it turned out in all epidemiological studies, the percentage of hemoglobin, fetal hemoglobin in the blood is one of the strongest predictor of, disease progression. And that actually led to the whole drug development area where people are really trying hard to, like find different ways to design small molecule drugs to increase the HbF. And the problem here is they don't know the mechanism, how this hemoglobin switching happens, what is like controlling it. So to only if we understand the mechanism, we can design the drug. But still serendipitously, like there was like drugs, anti-cancer drugs that they found that actually have an effect on the fetal hemoglobin. And even now hydroxyurea is the, like widely used drug. So that is how, like the HbF induction became a major, like mechanism to target to treat sickle cell disease. And like this is like people are looking at all different ways of doing, like in vitro screens and knockout studies. I was talking to Vijay Shankar, and, like he and his supervisor, colleague Stu Orkin, they were like pioneer in this field. So they were like working for a long time decoding this mechanism. So then this genome-wide association study happened, like it was also more serendipitous because they were not really working on any GWAS studies. That is the time the GWAS is just picking up. And then, like people from other groups actually, for some reason, they wanted, Gonzalo Abicazes, and he is like a big shot in the statistical genetics field. So he one day makes a phone call to, like Joel Hirschhorn, who actually does this statistical genetics course in Harvard, to ask, hey, we have this kind of study for fetal hemoglobin. We submitted to Science, and it was, like they said, we need a replication. Do you have anything? And that's how they got contact into,
Huh.
and they were like exactly waiting for such thing, right? So they wanted to know, they had no clue. He said we were nowhere close to identifying the transcription factor causing the hemoglobin switching. And then this happened, and then they collaborated, and then that's how there's like multiple publications. Two publications mainly happened in 2007 and 2008 that showed the clear signal in the BCL11A intronic region. And that's how the discovery happened. And then they picked up on that, and then there was like, they did the functional studies, and they kind of showed it's a erythroid specific, transcription factor that is mainly expressed only after birth, and it suppresses the hemoglobin, fetal hemoglobin production, and it increases the adult hemoglobin production. So that's like really incredible, like application of a GWAS. That's a kind of application we always, like look for, right? So looking at hypothesis free, looking at the full genome, trying to identify, in this case, trans regulator of HbF. So you cannot do that by case basis. There's like thousands of proteins, right? So that's a very clear application of a GWAS case.
Yeah, amazing. And I suspect probably the first of many, because we're seeing now every year as data sets get bigger, these kind of collaborations form. There's more and more of these interesting, not just primary discoveries of a hit, but also modifiers. And I think that was one of the big stories within this field that you looked at this year. There was a beautiful paper looking at a modifier of ApoL1, which is a risk factor for a relatively rare form of kidney disease called FSGS. We've spoken about APOL1 on the podcast actually before, and we'll have the author of this particular paper on in a couple of weeks, Alexander Bick, to go into this in a little bit more detail. But we asked them to join the podcast because I also thought it was an amazing story and piece of work. So maybe you can talk a little bit about a little bit of the backstory of APOL1 for those who aren't familiar, and then this newly discovered modifier that I think provides a really interesting window from a drug development perspective.
Yeah, sure. So ApoL1 is one of the other second most exciting, the story for me in this year. So I've been like following up on the story for a couple of years now. I remember I wrote a tweet maybe like last year or the previous year based on a New York Times article that really went, attracted a lot of attention. So ApoL1 is one of the, similar to BCL11A, ApoL1 is also one of the discoveries made very early. I think in 2007 or 2008, when the GWAS is just starting, people are looking at all the, trying to identify all the low-hanging fruits. And the special about this is like kind of a discovery that was made in the African Americans and African population. And So we know for a long time that kidney disease like disproportionately affecting African Americans. So, any kind of environment or, like systemic bias and things like that, structural bias cannot explain such a big difference. And always people thought there should be something genetics. And this is like the first one they identified in a very small sample size. It's a recessive association. It's caused by, two different uh, set of variants in APOL1. They call it like G1 and G2 haplotypes. And when they occur, like when it is like occurring in a recessive state, like both alleles are affected, it increases the kidney disease by odds ratio, I think like up to 16. And this is one of the main risk factor. And, and the reason why it is very highly increased in African population is, it's very similar to sickle cell variant increasing in frequency because of protection against malaria. You have this mutation. It's actually a gain of function mutation. It gives protection against a disease called sleeping sickness. It's caused by trypano, SOM infection, parasitic infection. And that's the reason this kind of increased in frequency. It's such a high level. And so this is like one of the very clear, target population, but the mechanism, people are working on it for like 20, 30 years, I think. The generally approved message is that this is a gain of function mutation. And so how will you treat it? You try to treat it by inhibiting the ApoL1, though I don't think like any kind of genetic evidence has been really came up. I mean, like ApoL1 loss of function mutations is extremely rare. And there is like, I think 6, 7 drugs already in the pipeline. Mainly they inhibit the ApoL1 inhibitors, small molecule and other modalities. But this evidence is surfaced this year. It's actually reported by two independent team, one from the Columbia University and one from the Vanderbilt University. That's where like Alex Big, who had this interview with. So they both reported the same protective association with the missense variant, which actually is a loss of function variant. When it happens, it kind of in the same G2 haplotype, it just eliminates the gain of function activity of the ApoL1. And for me, the most interesting part of this is like the origin of this mutation. So it was like happened, I think maybe like 10 years ago in a hospital in Ghana, in Africa, where doctors encountered a patient, like who had this trypanosome infection. And it's very rare to see those cases nowadays, and so they were like really curious and they kind of treated him appropriately, but also took the sample and they wanted to study, and they collaborated with the researchers in Belgium. And then the first thing they thought maybe this person doesn't have the APOL1 genotype. But to their surprise, they found this person has a homozygous APOL1 genotype, which is like a protection. It gives protection against this infection. So that kind of triggered them into looking into modifier variants. And they clearly identified this person as having this particular missense mutation, which actually is a loss of function. It completely abolishes the APOL1 activity. And that is what is now, people are reporting as a protective association in different population. And the beauty of this is this kind of validates the approach, right? So the approach is to inhibit the APOL1 and to treat the disease. And I think like already some of the drugs are very late in the clinical development. So hopefully 2024 might be the biggest story, will be like APOL1 drug approval, like we have for sickle cell disease this year.
Yes, no, absolutely. I think it's an amazing piece of work. And to me, part of the story, a big part of the story was that serendipitous discovery. And to me, it points to the importance of studying populations in countries that are outside of Europe. We know that we're not covering the genetic diversity around the world, even to a tenth of what's possible if we're so focused on the countries we're in today. I also think it points to the power of the scale that some of the very diverse cohorts, in particular in the US, are getting to. They did this analysis in both the Million Veterans Program and the All of Us, which are both have made a focus on recruiting a diverse set of Americans that include African Americans, people of Latino ancestry, and so on. And so the power of these large and diverse data sets has meant that you could make this discovery initially in a very small N of one study in Ghana, but then actually replicate it and provide a lot more robust statistics on a much bigger scale. So it's amazing to see these worlds coming together.
Yeah, it's really an evolving story, and there's like so many, exciting snippets in that. Maybe we can link the Twitter thread that I wrote about different, serendipitous part of this evolving story. Yeah.
Yeah, definitely. The role of serendipity in all of these, I think, is shouldn't be underestimated. I mean, the BCL11A story that you told involved a serendipitous phone call. This story absolutely does. The third one on your list, I don't know if we've got serendipity involved. There probably is, but it's another protective variant that we're looking at. So people may be familiar with the ApoE4, for which loss of function in a homozygous state confers a pretty marked increase in Alzheimer's disease and dementia risk. The heterozygous state does confer extra risk as well, though not to the extent. But this paper I also thought was really interesting. Was discovery of in a very large study, but a very rare loss of function variant in ApoE4 that chops off the E4 allele. And you wrote again a great breakdown on Twitter that we'll post a link to in the show notes. But it'd be great to just have your perspective on this. And in many ways, I mean, this is, again, part of the same story of using human genetics to validate drug targets that we're already going after, but also understand the mechanism in a lot more detail. So maybe you can talk about that one and what stood out to you about this particular study.
Yeah, so there's like two very closely related studies. One is that, was published this year. That's one I was talking about, the E4, loss of function variant co-inheriting with E4 allele. And one other, big story, I think 1 year before it was published in Nature Medicine, is like a Christchurch variant that is happening, that there's, there's like PSEN1 mutation causes very early onset Alzheimer's disease. And there is like a very big kindred in Colombia, right? That's where it was identified. And there they identified one woman who had this PSEN1 mutation, but never developed Alzheimer's, even at the late age. And then they found that this patient had a very rare missense variant. It's called like Christchurch variant. Was first reported in New Zealand, I think, like many years ago, in a E2 haplotype. But like this time they actually looked into the E3 haplotype, and so they kind of hypothesized that this missense variant in the ApoE gives protection against PSEN1. And so this is like a very extraordinary lab, just a single patient. And there was like one other development this year. There was a paper in Nature Neuroscience where they modeled the same Christchurch variant in an E4 haplotype in a mice. And they show when you do that, you can actually see a protective effect on the Alzheimer's pathology. And so Joy was like mentioning that is like there's like a really lot of interest in identifying these kind of modified variants. And the challenge is, the sample size. You really need a very large sample size. And for conditions like Alzheimer's, our sample size are still very modest. And so what you will see in these kind of papers, the other one is the ApoE4, where they, like scan through a big database of Alzheimer's patients who have been sequenced, and they kind of hand picked all the ones who had the peel-off variants in ApoE is like very rare. So people are wanting to see, what is this effect of peel-off on Alzheimer's risk. It's going to be very challenging, even in this paper, right? So one of the drawback of this paper is like it's a handful of patients, so it's kind of like make assumptions about what you see in this patient. I mean, they had very good phenotype. They had like biomarkers. They also have brain histopathology. One of the patients, or maybe a couple of the patients, had postmortem brain examination. And so they say that individuals who are heterozygote for E4 who co-inherited, the appeal of variation, right? That kind of chopped off the E4 haplotype. They actually did not develop Alzheimer's disease. This is not shown as like a statistical association, but it's like case by case basis. They explain it, and because they did not have the, like a lot of sample size, but still it's very interesting. It's kind of validates, I think, the current approaches a lot of companies are thinking about in terms of treating Alzheimer's disease is to use this ApoE4, like knockdown approach, allele specific knockdown approach and things like that. And it's yeah, it's very interesting paper, but we need to the association is not as strong as what you see in ApoL1, but it's a very important advance, I'd say. So some kind of positive, like evidence there to follow up.
Yeah, and you made the really, I think, interesting and important point in the tweet breakdown that something these studies don't tell you so easily is the when you have a knockout like this, a germline knockout, then you have essentially a lifelong knockout of this gene. But when you're designing a drug, you're very unlikely to have a lifelong knockout. And so there's a question of if you have an ApoE4 inhibitor, for example, at what point do you need to intervene to have some kind of impact that prevents or at least delays progression of someone developing Alzheimer's? And this is a topic area I'm really interested in. We have a guest that episode will go live in January, but who's doing this kind of work in ALS and in genetic forms of ALS and MND, Michael Benatar, who's at Miami's Miller School of Medicine and has been for the past 2 decades studying people who carry genetic risk factors of ALS and measuring very carefully biomarkers, both genomic and otherwise, that correlate with progression and phenoconversion ultimately to ALS and MND. And I think it's going to be one of the most interesting, hopefully, changes of our time where we can start to flip from treating symptomatic patients in neurodegenerative disease to treating asymptomatic patients and preventing this from ever happening. But I think certainly in that field, but also in Alzheimer's, it depends a lot on identifying good biomarkers of early progression so that you can identify at what point is the right time to intervene with that medicine that hopefully isn't when somebody has developed a full course of disease, but is at the mild cognitive impairment stage or even earlier on if there's good biomarkers to detect that.
Yeah, that's a really great point. That's one of the fundamental challenge of Alzheimer's trials. At what point you need to intervene, right? So, but also it's kind of something that's also we often get to, like questions from our drug developing colleagues, from who does all this preclinical studies, like, so what we can get from the human genetic evidence is like a lifelong knockdown or, like
Yes.
whatever. So, but we cannot get any kind of evidence where you can, like if you knock down a gene only in the, like in the postnatal period, or maybe for a very good example is like the CHRNB2 that I've been like, working on it. So, so like, there's like in a typically for a smoking treatment, right? So the Chantix that we use prescribed for smoking cessation is just only for a short duration until the person quits smoking.
Yeah.
Then, then the person can get back to their life, right? So basically, if you have to design drug, like something like Chantix, then you have to like probably, inhibit the gene or something for a very short duration, right? And this is kind of, if you put this in the context of not having this gene, or, like having a dysfunctional gene for a whole life, then really we need to consider about these differences when we interpret the adverse effects as well, right? So
Yeah.
there are a lot of situations where, a gene is like very essential during the neurodevelopment, I mean, brain development, but later not. Or also like it can be very lethal only when it is like happens for the whole, lifetime, but not when you do it in the very specific time point. And this is like really difficult to get from a, human genetics evidence. Only like that's where like really the preclinical studies and animal models and things like that can really help. So yeah, that's a really great point. So I think it's even a bigger challenge in Alzheimer's trials.
Yeah, we're starting to move into rare disease a little bit now, and theme two maybe is the transition from common disease genetics and their role in drug development to rare disease, and especially as the number of rare disease focused programs and the scale of some of these population scale biobanks grow, we start to get really detailed looks into rare and ultra rare populations. And looping back maybe to the BCL 11A story that we talked about at the top of the show around the sickle cell approval, there was an interesting paper that you highlighted that looked at this from a very different perspective in a neurodevelopmental disorder cohort and tied into that story. Maybe you can talk a little bit about that.
Yeah, so this is another theme that I really, like. So, like how studying rare disease patients inform common disease pathophysiology, also drug targets for common disease. And so after we briefly touch on the BCL11A, I will talk about this HMGCR, the statin associated rare disease. Is one of the, like famous story this year. So in the BCL11A, what happened is like, so one of the ways like the fundamental problem with the GWAS studies is like you identify variants, non-coding variants, right? So for example, for BCL11A, it's an intronic variant within the BCL11A, and then later they kind of studied further and then found that this is located within enhancer region, right? So it's very difficult to identify the causal gene. We'll have this FTO story coming up. So one of the ways you can identify causal gene is to, like you can delete the gene in animal model, or you can find individual humans who naturally lack this gene. So that's where this rare disease patients come up, and then they can give you, like either they can help you to, okay, eliminate this is not the causal gene because you don't see this phenotype in the patient, or maybe you can help them to confirm. So similarly, this BCL11A is the gene, like, but they wanted to, people like they really wanted to know what happens in individuals who have haplo, like mutations, germline mutations in BCL11A. So it turns out BCL11A is like a neurodevelopmental gene. People who have haploinsufficiency have like intellectual disability, and it's like a lot of brain related, developmental issues. But when, like when they actually started looking into the literature, I was talking to like Vijay Shankran. So they actually was like looking into this patients. Did they anything in the clinical reports? Did they report anything on the hematological phenotype? It turned out that in the same year when they actually published the GWAS finding, there was like another, like coincidental paper reporting on micro deletions in 2P regions causing neurodevelopmental syndrome. I think that's the first one, right? At that point, they did not have any idea what's the, like it's the mainly BCL11A. But if you look into the reports, they reported everything, like lung phenotype, like all the brain phenotype and circulation and everything, but they did not report anything about hematological. So they were like,
Huh.
that's one piece of information that was like missing from that paper. So they have to like painfully wait and collaborate with the rare disease researchers in the Harvard and collect patients, right? So there's like recurrent microdeletions in this region. They have to identify a patient where the deletion is smallest possible, so it only hits the BCL11A. And finally, they found 3 patients, and then they looked into the hemoglobin fraction, and it's like more than 20%, 20 to 30%. And this kind of gave an important insight in the drug development, because the people want to know what level you have to induce the hemoglobin fraction, like fetal hemoglobin, to achieve, like some kind of clinically meaningful outcome. From the natural history study, they say it's kind of 20% ballpark. So how much you have to knock it down the BCL11A. So you cannot like completely knock down the BCL11A, because it seems if you do that, it's kind of some toxic for the B cells and things like that. So you need to dial it down. And 50%, that's what these haploinsufficient neurodevelopmental patients have, right? Gave an insight. If you dial it, if you reduce it by 50%, you can increase the HbF by more than 20%. And that is enough to cause, like a clinically
Yeah.
meaningful outcome. It's like absolutely incredible example of how a rare disease can give, like inform drug development and also establish causal genes in the GWAS locus.
Yeah, amazing. And I guess this actually maybe dovetails really nicely into the follow-up story within this theme of studying rare disease to inform some of the common disease findings. But in my mind, it makes me wonder whether with these new approved therapies going after BCL11A, whether some care needs to be paid to what's happening on the other allele that doesn't get edited. So if people are carrying modifiers that are already reducing their expression to some extent, that this edit pushes it not to 50%, but to something that becomes toxic. And I think, maybe this is a good opportunity. We don't know the answer to that question, I don't think, but we do know the answer now to one of the really interesting puzzles around statins. So you again had a great thread on this that I learned this reading the thread, but that statins have a common side effect of myopathy. And this was a little bit of a puzzle for a long time until this piece of work was done. So maybe you can talk a little bit about the work and how they managed to crack the code on why this incredibly common approved therapy that millions, hundreds of millions probably around the world take today has this side effect that we still didn't understand until relatively recently.
Yeah, so this is another fascinating story. So this actually stemmed from my interest in the HMGCR because I don't know if you came across such discussions. There's often we have this intense discussion about effect size, there is like one group who says GWAS variants have a very minute effect size.
Yes.
What are you going to do with this locus? Like there is
Yes.
another group that says they are common variants, right? You can actually, you can increase the effect size substantially by by using different modalities, right? But the important point is it safe to do that? We need the human genetics things. So I was always interested in what is the rare variant associations of HMGCR. And this is when I actually came across this paper in American Journal of Human Genetics, where they first time reported this biallelic mutation HMGCR. And it turned out there was like a previous paper that was published, same findings, same similar findings in PNAS from an Israeli scientist in January. So both of them report the same thing. So they identified biallelic mutations as the cause of a very rare, myopathy, like it's called limb girdle disease, and it's characterized by very severe proximal muscle weakness to the point, like it starts around 40 years of age, and then people become like, they get in the wheelchair, and then they had to get assisted ventilation. It's a very severe disease, and they found in this like in two families, they identified the causal mutation. But the most fascinating part of this is like, this is what like kind of driven me to look into the origin of the statins, right? So we should link this, this article about the person Akiro Endo. He's a scientist who actually discovered the statins. He's a Japanese scientist. It's really fascinating. Like, so this is also a serendipitous kind of discovery, maybe. So he's like really fascinated about Alexander Fleming and penicillin, like discoveries. So he was working on the fungi, different fungi, and how they kind of protect against ward off the infections. And it turned out they kind of produce very specific chemical compounds that interferes with the cholesterol synthesis, very important for this pathogens infecting. And so he started studying in that, and then he was like he's like collecting compounds from different species of fungi. And there was like one time he got this rice from a local grocery store in Kyoto, and the fungi that he extracted from that have this $1 billion jackpot
Wow.
compound that kind of led to the whole statins group or family of compounds. He never got like the credit. I think he worked for a company called Sanctuor or something, and then but later I think the Nobel Prize, when they awarded Nobel Prize for the cholesterol pathway, the scientists actually said it all the patients who have benefited from the statins owe to Akira Endo. So the interesting part is like when he actually designed the statins and dosed the first patient, it was like 18 years old female girl. they actually gave a very high dose, and this girl got a severe proximal muscle weakness, and they had to like stop the trial. And somehow they kind of fought against the company and the regulations and continued the trial with a lower dose, and it became successful.
Wow.
But that part of that particular finding of severe muscle weakness in that first patient kind of buried in the literature, I think. And so, but later it turned out one of the common side effects is muscle pain. But there is a very big debate in the clinicians who actually prescribe statins. You say like a lot of them don't believe that this is real pain. I mean, there are even clinical trials where you say you prescribe statins without saying you will get muscle pain, right? Anything, and they don't get muscle pain. But
Huh.
this kind of the first time we have a like a genetic evidence where we say if there is a loss of function mutations in HMG-CR, then you get like severe myopathy. And the beauty of this is like the patient, it's researchers from the Israel who did this, they also continue to do a N1, N is equal to 1 trial that, so the problem is like this HMG-CR actually can, like this it reduces the HMG-CoA to mevalonate, right? Which is the precursor of cholesterol synthesis. So the problem is like because there's a mevalonate deficiency. So they thought, okay, if you supplement mevalonate in these patients, will it have an effect? And they kind of did this mevalonate supplementation, and it had a dramatic effect. In like in day 21, the patient they treated regained a lot of muscle strength. She was able to breathe without ventilation, and this is like really amazing. And they kind of see that this could be melanate supplementation could become like a standard way of treating severe myopathy associated with statin supplementation. So I think this is like an incredible example of a rare disease, very rare disease, right? So informing a drug that is used for a very common condition. So yeah, that's
Yeah.
like I really like the story a lot. Um,
So is the idea and is the idea that this supplementation with I'm not going to remember the name of this specific enzyme that they supplement. Yeah, would the idea be that for people who are on statins and experiencing either mild or moderate or severe muscle pain, they might be able to be prescribed this to reverse that aspect of the phenotype? Is that right?
Yeah, that's the speculation, but it's not really clear because you're basically reversing the effect of your drug, right? I mean, from what they see in these patients, the mevalonate supplementation did not change the cholesterol levels, did not influence the cholesterol levels. So there could be more than what we know, just from this. What we know is like the simple chemical reaction that is like blocking the cholesterol synthesis and things. Maybe there are like more, like alternative reactions and things like that. So it's more of a speculation, but I think it's like the insights that we got here from this rare disease patients is really great. And even for not for statin associated myopathy, I mean, this malnet supplementation is a very simple treatment for very rare disease. Imagine kind
Yes.
of treatment we design today for rare disease, CRISPR treatment, things like that for like $2 million. This is very simple, supplementation. That's like it's like getting back their life for them, right? So it's like incredible.
Yes, absolutely. And I think with these kind of a couple of the discoveries we've talked about today are kind of happening in order, in the sense that BCL11A is discovered, the mechanisms worked out, and then drug development starts to happen probably off the back of some of those discoveries. But then some of them are in completely garbled up order, like the drug development is years underway for APOL1, and people more or less just figure out ways or trust that they can safely inhibit it. And there's, I'm sure, good in vitro models or okay ones that, but these kind of things do make me wonder about the path dependency of some of these development stories, where if this particular relationship with the with the myopathy phenotype had been more thoroughly studied earlier and not kind of buried and with the work continuing in secret or in a gray area, would they have designed it differently? Would they have chased this down? Would we have never had the discovery in the first place? It's hard to know, but it's really interesting to hear some of these stories that are almost coloring in the lines retrospectively, and these things have happened anyways, but others that are really the reason why some of these programs are starting in the first place.
Yeah, so like I just need to add a comment here. This is one of the things that we kind of, like as a person working in drug discovery research in a pharma company, is like there's like two different areas that we are focusing on. One is like target discovery, right? And the other one is like we use human genetics to try to validate the ongoing programs.
Yes.
And often, and that's like not as often fruitful as it is for target discovery, but like people always come with, hey, did you find any human genetic evidence for this program? And like often they are very constrained genes we don't find, but sometimes this kind of human genetic evidence kind of informs you, you might find this adverse effect, or you might see that the efficacy is not as great you would expect, because the maximum thing we see in patient who completely lack this gene is only this much, so you cannot push it further. So insights like this can really help with the ongoing drug program. And that is one of the reasons a lot of companies are now starting to establish their own human genetics team to, support the ongoing drug programs.
Yes, and if people are interested in a deep dive into this, we had David Ochoa from Open Targets on episode 112, which was just out, I think 2 episodes before you'll be hearing this one, and we have Matt Nelson from Deerfield in the very next episode after this, so you can hear both of their perspectives on a really macro level of how genetics plays a role in drug discovery development, and also increasingly clinical trial stoppage, success, and so on. It's an interesting time with some very great perspectives on this.
Yeah, I listened to the David's episode like maybe 2 weeks ago. It was really great. Yeah, looking forward to listening to
Yeah,
Matt. It's
he's very
interesting.
good. I think they probably agree on most things, but maybe disagree on some. So it'll be a good pair to listen to. I think moving on further into the mysteries of how we actually understand these GWAS loci. I think this example that you pull out here is an amazing one, because it's one thing to discover a locus and think you understand it, but I think this particular story just underscores how complex the biology can be, even when a gene is so incredibly well studied, like the FTO gene and locus is. Sometimes it takes some amazing work and some very detailed unpicking of what the heck is going on to figure out what the heck is going on. So maybe you can talk about this. I think this is probably one of your more interesting deep dives that you've done this year, and I think was very well received. 300 something retweets and lots of people interested in it.
Yeah, it got a lot of attraction. So I think people were really interested in this whole FTO story. I mean, I chose this example to highlight, like often we get this question of, we have like common variants versus rare variants, which ones, like are more useful for drug development. And it's, like mostly the companies go after rare variants, right? So they do exome sequencing. And common variants, it's the challenge associated with the common variant is like to identify the causal genes at the locus. And for the rare variants, our starting point is gene, so you can just proceed with, like for some, if you start with a common variant, probably you'll have like 10 years or so just figuring out what gene it is. And FTO is like a fantastic example, I think. So like it again falls under the same theme of low-hanging fruits, like BCL11A, ApoL1, FTO, they all been discovered in the same, almost very close, 2007, 2008. So FTO was the very first association identified for obesity. And interestingly, it was first identified as an association type 2 diabetes. A lot of people, I don't think they know. So they found this as an association with type 2 diabetes based on a case control analysis from the Wellcome Trust Case Control Consortium. And then they kind of adjusted for BMI and to see, and this is like completely mediated by BMI. It's a fantastic paper. So one of the landmark paper, I think it was a science paper in 2007. So, but the thing is like, they didn't have like any kind of idea what is the causal gene. I think again, it's an intronic variant. And so they kind of, believed FTO it is. And like this finding really triggered a lot of research. There's like many landmark publications you would call, or like big publications like Nature Science, NEJM, like step by step decoding this loci. Like the very first obvious thing to do is like, okay, if FTO is the causal gene, let's delete this gene in mice and see what happens. And that's what they did. And they found that when you knock out the FTO, the mice is like developmentally stunted and it has a lean body mass. And so they thought, okay, so then FTO is the causal gene. It has an effect on the, like the body weight. And then immediately there was follow-up studies that showed if you knock out FTO after, the animal is born, right, in the adult stage, you don't see this effect, what you see. So it's kind of a more developmental effect. And you also see if you knock out the FTO only in the adipose tissue, right? So so that kind of added some confusion to the story. And then we had this kind of big two major publications, side-by-side publications in 2014, I think. One was in Nature, another one was in NEJM, that kind of showed that the causal gene is not FTO, but it was like a couple of genes located a few kb away. And the mechanism is that this intronic region in the FTO acts as an enhancer for these two, IRX1 and 2 genes located far away. And then the 3D space, they kind of, like fold and go and touch with the promoter. And this is often kind of like a very common example to show whenever discussion comes up, closest gene is the causal gene for GWAS locus. When you want to look at the exception, they often highlight this as an example. And then I think then there was like follow-up studies. Then again, it showed when you actually delete the intronic region within, like this, because all these studies have been like doing FTO knockout or something like that. It's like you have to like exactly delete the region where the GWAS signal is done, right? So then if you delete the intronic region, again, you can see it affects the expression of IRX1 and IRX3, 2 and 3, and they kind of believe that these are the causal genes. Then this year, we had this paper in Nature Metabolism from scientists from China, who argued that if you want to really, like mimic the human GWAS finding, then you have to take this exact variant and knock in the mice and see what happens. And they actually did that. And they found that the effect was completely opposite to what they see in the humans. In the humans, the homozygotes actually weigh 3 kilograms more than the non-carriers. So it increases the BMI. And in the mice, actually decrease the BMI. And it does so by increasing the basal metabolic rate and brown, adipose tissue thermogenesis. And so this kind of add to the confusion. And, like they also showed this knocking in actually affected the FTO expression, but not the IRX1, IRX2 expression. So they came back to the
Right.
causal gene being FTO. And the most interesting part of this is like they kind of show, they kind of looked into the ambient temperature, right? So when you are studying about the brown tissue thermogenesis, adipose thermogenesis, so the ambient temperature, what they actually did this experiment in the mice is like is ambient for humans, but not for mice. So mice actually, like their ambient temperature is like more than like 30 degrees Celsius or something. So they changed the temperature, repeated the experiment, and all these effects got blunted. So there's like two factors it seems important. One is presence of brown adipose tissue, and the other one is the ambient temperature. And the first factor is a big difference between mice and humans, because in the mice, they have the brown adipose tissue even as adults, but in humans, we have it only during the infant stage.
Mm.
We don't have it in adults. And it turned out there is a paper which actually showed the FTO variant actually decreases the weight when during the very early stage of life, and then it starts to increase. And so this kind of explains why this opposing effect is seen in the early infantile stage, because that's when we have the brown adipose tissue. So I think still there is like a lot of
Yeah.
unknowns, but the development is, I think this is one of the most studied GWAS locus, I think, in the literature. And still, I don't think there is any kind of agreement which one is the causal gene here. There's like people
Yeah.
who will call FTO, people will say ARX1, 2, 213. So so
It's very humbling, isn't it, to study a gene for or a locus for this long and still feel like your world gets flipped upside down every time a new major paper comes out, because like you said, they found the exact opposite of what they were. But I was struck by how meticulous they were at chasing this down with looking at temperature changes, the beautiful graph that you have in the tweet thread that shows this inversion basically of the curves early in life and later in life. It's I remember seeing a talk many years ago where somebody walked through the science of I don't actually remember the gene or the therapy anymore, but they walked through about 20 years from discovery of the monogenic condition through to a therapy being created, and some of these twists and turns along the way. And I think your GWAS storytelling career is going to have a very long life, given how many of these things we need to figure out.
Yeah, I think there's all this early, what you call is like low hanging fruits are the ones that like often you're, like very difficult to crack it open. So,
Yeah.
like most of the ones that we discovered in 2007, people discovered GWAS, still it's that where people are trying to make sense of those loci. So the literature will evolve.
Yeah, we're perfectly on time here to cover our fourth topic in the first half of this episode, and then we're going to take a little break and come back for part two. But this last theme is about genetic drift, endogamy, consanguinity, and also more generally the rise of large scale studies and very carefully designed studies looking beyond European populations. So maybe we could start with the FinGen flagship paper publications, or multiple of them in Nature. It'd be great to talk a little bit about FinGen. I haven't actually had anyone on the podcast from FinGen, and I'll do that because I think it's an incredible study. But maybe you can talk a little bit about that, and then follow up with the 23andMe paper, where they found a really interesting founder variant in a Puerto Rican population, to talk a little bit about the role that these founder populations play, very important one in human genetics.
Yeah, so this is another favorite theme of mine. So looking into studying populations outside Europe, like non-European populations, also studying, like looking outside the outbred population, right? So one of the two great resources, like we know Icelandic population, Decode, is one of they are the global leaders, and they pioneered this research, and they're kind of an isolated population. And similarly, the FinGen recently evolved, it's also an isolated population. It underwent multiple bottlenecks. And the result of this is like there are a lot of rare variants that, because of genetic drift, they rise to very high frequency. And we know this, like there's like so many interesting publications from, like isolated studies from different parts of Finland. So there's like we also know a lot of diseases, recessive diseases that are, like common in Finland. But then this FinGen was like established, I think maybe 5 years ago or so. And then they kind of made this 500,000, very similar to UK Biobank, establishing a biobank in FinGen based on, like health, incorporating with health system. And this one is the first flagship paper from the first release where they see genotype. They did not sequence. So that's another interesting part of this is like, so when you actually have this kind of founder populations, right? So there's two things when you have this bottleneck population. One is there are a lot of rare variants that go drift into very high frequency. And these are the variants that you can actually identify using genotyping and imputation. And the second thing is there won't be, like compared to outbred population, the rare variants like singletons or other variants are very, like less because of, lack of diversity, right? So one of the things that you will notice that neither Iceland, right, or Finland, they did not go for like sequencing the whole population. So they had this formula of sequencing a subset of the population and deriving haplotypes from there and then imputing it. So that is how they did this, but probably they will change, like as technologies and prices come down. For now, this paper, flagship paper, is based on genotyping of 220,000. And so they have like two parallel publications. One they did, looked into they did a GWAS association studies for all the electronic health record derived phenotypes in this population, and also like meta-analyzed with UK Biobank. And so the major message from here is like there are many associations that are unique to Finland. And these are the ones that are driven by drifted variants in Finland. So these variants are multiple times enriched in Finland and absent elsewhere. So this is one of the importance of studying isolated populations. So to find same association in UK Biobank and other population, you will really need like to sequence millions of individuals to capture that handful of carriers. And if you go to Finland, you will find like 100 or 200 carriers. And they have like many interesting, associations. I think they highlight one of the most interesting one is an association with inflammatory bowel disease. There is like a TNRC18, and it's like 116-fold enriched in the Finland
Wow.
population. And the risk odds ratio is almost same, very same as the NOD2, the major risk factor inflammatory bowel disease that we know for a long time. So, very similar risk factor, but it's been hidden because people carrying this mutation were only in the Finland. And that's like really fascinating. And so in many similar examples. There's like the first paper where they show unique associations in the Finland from drifted variants. And there's like a parallel publication where they focus on recessive genetic associations. So this is really an evolve, area where emerging databases, biobanks are really giving insights into our understanding of Mendelian diseases. So we know autosomal dominant disease, recessive disease, all of this, our knowledge about these diseases come from isolated families and patients from, the clinics doctors have seen many, many years ago. And like, so this is the first time we are able to get like like their validation or maybe replication of these so-called recessive associations in a population scale. And there's like heritage recessive diseases like hearing loss, retinal dystrophies that are already known to be common in Finland. But they also, when they did this systematically, they also found a lot of associations that was significant only when you do a recessive model. They highlight few like they have CAPS CAP7, I think it's associated with adult onset cataract, and there is like a female infertility gene, some gene related to estrogen receptor binding antigen something, and it's very, very interesting, associations. Seems they are recessive diseases that we never knew about it before. So this is kind of, it's a flagship paper and kind of highlights the importance of studying founder populations, which is very important, right? So there are so many such populations around the world, and we don't know. We are just starting to, learn about them. So that
Yeah.
kind of,
Yeah, and it's almost, you've highlighted the double importance of these studies. It is important to understand the diversity of genetics around the world, and it helps us from a fundamental discovery perspective, but it's also really critical for healthcare systems in every country to understand actually what are the differences in the the particular genetic diseases that might be at either at higher rate or completely non-existent elsewhere in the world that do exist in your country. So I think we're going to talk through quite a few examples in this category, but I think it's a really important double value of investing in these kinds of programs, that you both get to further the science while also making sure that the discoveries are really localized correctly to the group of people that ultimately are trying to help by bringing this to the healthcare system.
yeah. So one other thing I wanted to highlight here is like, FinGen is like they openly share their association results, and there's been like incredible resource for both academic researchers and as well as, people from the pharma industries, right? I have one example from my own case where I did this association. We found the CHRNB2, right? So we found that this rare variants, one missense variant is the one actually was like the main driver of this association. And this like incredible, it's very rare it's only seen in the European population, but it's rare in the, non-Finnish Europeans, but it's like almost 10 times enriched in the Finland population. So just looking up this variant in the FinGen database kind of, showed that, hey, this is enriched. It's like there are more carriers there. Even looking at a very small subset, we looked into like close to 800,000 in our sample, but in the FinGen, it's only like 100, 200,000, but still we were able to replicate the protective association, right? So this is a lot of people don't realize the value of this database. I always say in the Twitter, never forget to check FinGen. And if you find anything, first thing you should go to the FinGen, browser and check for, like results of your favorite gene there. You will be pleasantly surprised often.
Yes, maybe I'd love to just finish this first part with the 23andMe story from Puerto Rico, and then we can pick up in the start of the next episode talking about some of the other findings and studies that fall under this broader category of looking at non-European populations, and in particular the concept of endogamy and consanguinity. That's a really important one, and is again both driving important discoveries, but also helping us to make discoveries that are more localized and relevant to the particular populations around the world. So maybe you could finish with this really interesting study in Puerto Rico, and then we'll take our break and come back for part two.
Yeah. So this is like a paper from 23andMe. So this kind of falls under the theme that, like often these founder populations or endogamous populations, you don't really have to like travel to all these exotic areas to actually do the studies. You have opportunities to make these discoveries, even the databases that we access day to day, right? So this is one example. There's also other couple of examples from the Mount Sinai Biobank and also from the UCLA Biobank. But here, like they kind of 23andMe researchers looked into the genetics, like genetic associations focusing on Puerto Rican population, Hispanic and Latin American populations has been like historically underrepresented in genetic studies. And so I'm kind of more interested in all the evolving discoveries in this population, and this kind of really stood out. So Puerto Rican population is one of the, the Caribbean island population. It has undergone multiple bottleneck events because of colonization and things like that, that actually resulted in a strong founder effect. So this region is very famous for a syndrome called Hermansky-Pudlak syndrome. It's a albinism. It's very, very prevalent. If you happen to go to Puerto Rico, it's very hard to miss. You will see almost like one in, every 10 person you meet will be like an albino. So this is like a disease caused, like the gene was identified only after they learned about the disease. So it affects your skin and it affects your eye. People go blind sometimes very early in age, and then people have bleeding problem, and also people have like lung fibrosis. There is great coverage about this, problem in Puerto Rico. So they often compare this, pulmonary fibrosis condition as a ticking time bomb in the chest. So you don't know, like people sometimes who just travel on the road, just catch their chest and then fall down and they just die. So it's very common condition, and so this is like known for a long time. So it's like my interest is like what other, common conditions that we have because of this kind of founder effect and things like that. There's like few examples, and the recent one was from the 23andMe, where they identified like a gene called ITGA6, where heterozygous loss of function variants are associated with a huge risk effect on adult onset cancer. I think odds ratio is like 12 or something, and also it has like a impressive association on the age of onset. 13 years earlier, people who are heterozygous for that. And interestingly, this is like a dominant associations, and the recessive association has a very different disease, like some blistering of the skin, some other conditions. So I think it's some laminin associated gene or something. It's very important for the cell surface adhesion and integrity, and this kind of in some way affect the lens fibers, but also when it happens like a complete deficiency, it also affects the skin. So it's very interesting, and like probably this is one important, and we didn't know all these years, and just using a very small set of population from the 23andMe, they kind of caught this gene, this association. This is like really very interesting finding.
Yeah, I think that's a great one to end on. We've covered a huge amount of ground here, a number of both stories that reached a conclusion in the sense of they were discovered and then ultimately became approved therapies, although that story is still going as we try to understand how that drug is going to be priced, how access is going to be made available to the millions of people around the world who need it. We aren't going to cover that in this episode, but next part of this that you have to look forward to, we're going to continue the discussion around endogamous and consanguineous populations and some of the important findings this year. We're also going to talk about some interesting, very early stage rare variant discoveries and some other GWAS discoveries, and finally finish up with some of the highlights around complex trait genetics, some really interesting stories around tandem repeats, which long read sequencing and larger scale whole genome sequencing are starting to shed some light on some of the last parts of the genome that we haven't been able to read. And then finally, we'll finish up with some predictions for the future. I think quite a few of your predictions last year came true, in particular one about these non-European population studies being a really big part of the story this year. So stay tuned with us. We'll be back with part 2 in a separate episode that will be released a day or two after this one, just so we don't hit you with a 3-hour episode in one go. But thanks again for listening, and we'll see you for the next episode.
Thank you, Patrick.